Click for IB Biology. Here we're looking at both the technique of gel electrophoresis and some of its applications in genetic techniques. There's a blank PDF of this presentation down in the description down below this video, along with 10 questions which have answers so that you can check your understanding of this content. And the channel links, of course, will take you to the main channel and indeed to the playlist so that you can look at other videos in this sequence. Gel electrophoresis. The students would recognize this technique if comparing it to a form of chromatography, the separation of different components. This technique relies on the use of gels. There are a number of different gels which may be selected depending upon the particular separation that we're attempting to do. But for instance, agarose gels are very commonly used in gel electrophoresis. Wells are cut into the gel, which is sitting in a buffer solution. So we would imagine that we have fragments of DNA of different size, and these are introduced to the wells at this position here in close proximity to the negative electrode. An electrical field is then applied across the gel and the buffer, with the negative electrode at this end and a positive electrode at this end. The DNA fragments will migrate through the gel and separate on the basis of their size and of their charge. It is worth noting that DNA normally has a negative charge, and so they're attracted towards the positive electrode. So the fragments of DNA will separate on the basis of how large they are and the charge that they're carrying. Once this technique is complete, it is then possible to identify the fragments either by using staining techniques or by using radioisotopes. In this way, we're able to see the different fragments within the sample. Large fragments will not have traveled very far, and small fragments will have traveled a long distance, as is shown in this particular image here. Let's look at some applications of gel electrophoresis. A child has inherited a chromosome from its mother and a chromosome from its father to form what is actually called a homologous pair. In the chromosome, in the non-coding regions, sometimes called the nonsense regions of DNA, there are what are called satellite tandem repeats, STR. And in different individuals, the number of STRs is different. So we can see here that the mother has 5 STRs in this region here, whereas compared to the father's chromosome, it has 2. If we then introduce these to gel electrophoresis, the mother's STR, being larger, travels a relatively short distance through the gel. The father's, on the other hand, being smaller, only composed of 2 STRs, travels this distance, a larger distance, through the agarose gel. If we then think of the child will have both the mother and the father's chromosomes and will have both components. So if we chop out the STR from here and from here, put the samples into the well, when turning on the electrical field, we will find that we have the mother's STR and the father's STR, showing that the child is, the child's parents are this one here and this one here. Let's look at the application in more detail. Here we have a paternity investigation, for which we will have the mother's DNA, the child's DNA, and then we're trying to identify whether male 1 or male 2 is the father of this child. Any band which is present within the child will have to be present either in the mother or in the father. So if we come to this level here, we can see that this band is present in male 1. We come to this position here and we find that this band is present in the mother. Just below it, we can see that these two match up, showing once more that male 1 appears to be the father. If we come down to this position here, we find that the child's band here, this piece of DNA, is also present within male 1. And so we have an increasing number of markers within our paternity investigation which are suggesting that male 1 is the father. And this is a crucial aspect of gel electrophoresis with these investigations, is that we need to increase the number of markers to reduce the chance of any false positives. Notice that male 2 has no matching bands with the child. So in this particular case, this being the mother, this is the child, and male 1 is indeed the father. Within forensic investigations, we have in our comparison here, we have a victim and we have suspect, we have a specimen DNA which is taken from the crime site, and then we have 3 suspects labeled here as 1, 2, and 3. Now, of course, the purpose of the victim's DNA sample is to eliminate their DNA from the specimen, which could have happened by contamination. And so as we go through the comparison once more, notice that the specimen from the crime site matches this one here, suggesting that suspect 1 may have been at the crime site. Note that it doesn't show a crime has been committed. What it shows is that suspect 1 may have been at the crime site. But as we continue our investigation, we start to note that other specimen fragments of DNA continue to match up with suspect 1, but not with 2 and not with 3. And so once more, we have a large number of markers indicating that suspect 1 DNA matches the specimen, suggesting that suspect 1 left DNA at the crime site. Notice that this technique relies on the amplification through PCR as before the gel electrophoresis can actually be carried out. The samples at a crime site are small, and they are amplified through DNA polymerase techniques so that gel electrophoresis can then be used to find the associations. The links appearing here are back to the main channel, taking us on to further aspects of the course and indeed to the other videos in this playlist.